Hi, my name is Shania Pintor,
And my name is Lesly Shania.
and we're in Biology 140606, and we are going to be talking about...
The differences between aerobic respiration and photosynthesis.
Okay, so in life there's three types of respiration: aerobic, anaerobic, and fermentation. And in this first one, we're going to be talking about aerobic respiration. So the first process in aerobic respiration is glycolysis, and glycolysis happens in the cytosol that's outside of the mitochondria. And you can start with any molecule. You can start with fructose, glucose, sucrose, and in this example we're starting with G3P. So G3P stands for glyceraldehyde triphosphate, which means it has 3 carbons. And in this process, the carbons are getting cleaved. So when the carbon gets cleaved, it creates NADH, which gives you the energy to create ATP. And it does this until it creates 1 ATP and 2 NADHs. And that results in pyruvate. So that's with oxygen. And if there's no oxygen, it actually, pyruvate turns into lactate in a process called fermentation. So the extra ions are going onto the pyruvate, and pyruvate becomes lactate. That way you can run away from the bear that you're being chased from.
Okay, so before you can go into the Krebs cycle, you have to go into a half step where you take the pyruvate from glycolysis and cleave off carbons to make acetyl-CoA. And then from there you can enter the Krebs cycle where the acetyl-CoA binds with oxaloacetate to make citric acid. And that is a process called substrate phosphorylation. This all happens in the matrix of the mitochondria. And the end products are 3 CO2s, 5 NADHs, 1 FADH2, and 1 ATP.
Okay, so the third and last step of aerobic respiration is the electron transport chain. So this happens in the inner membrane space of the mitochondria. And in the electron transport chains, you have 3 proton pumps. And these 3 proton pumps are called a proton gradient, or a chemiosmotic gradient, or a pH gradient, or an ion gradient, and they all mean the same thing. But that's the flow of electrons. They're dropping in quanta, and they're moving throughout the 3 proton pumps. And when this happens, they actually create hydrogen ions, which are released out. And in the end, the hydrogen ions, do you want me to say over here? In the end, the hydrogen ions, there's so many in there that they have to get out, diffusion, right? So they go through and they power ATP synthase, which is an enzyme, and that's where ADP plus P creates ATP in a process called chemiosmosis.
So next we have photosynthesis. In photosynthesis, it starts with the sun releasing photons, which are captured by the antenna complex in the thylakoid, and then funneled into the reaction center, where it excites the electron, and then it drops in quantum, and then releases a hydrogen ion into the stroma, which powers ATP synthase to make ATP.
In the other photosystem, photosystem 1, the electrons again get excited by more sunlight, more photons, exciting the electron and moving it up. And then it continues to drop quanta, and this creates NADPH.
Next, we have the Calvin cycle. In the Calvin cycle, rubisco binds with carbon dioxide and RUBP and goes through carbon fixation to make 6 G3Ps. The 6 G3Ps reduces into 1 G3P and 5 G3P. The 5 G3P are used for the regeneration of RUBP and continues the cycle. In hot or dry climates, the stomata closes up to prevent water loss, but also causes oxygen buildup, okay, and which is bad because that causes the plant to die. So plants like C4 plants have adapted to take the CO2 into the mesophyll and cleave off a carbon to make oxaloacetate. After the oxaloacetate is cleaved off, or made, it follows the steps to make PEP carboxylate. The PEP carboxylate goes into the bundle sheath cells and breaks off to make CO2, which is used in the Calvin cycle in the bundle sheath.
So the other type of plants that have adapted for this are can plants, and those are your succulents, so cactuses and things like that. So what they do is they do carbon fixation, but they do it during the nighttime. That way the stomata can be open without it being hot or dry. And then when it does become hot and dry, the stomata closes, and that's when the Calvin cycle occurs. again.